Okay, this is part 5 of the muscular system. Sorry, I apologize the way part 4 abruptly ended, but I was kind of done saying what I had to say. So I'm going to get right in. We have quite a bit to cover. So for skeletal muscle contraction, there are 3 things that have to happen, and we've already covered some of them. Okay, we have covered that we need calcium. Right, we know what calcium does. It binds to troponin, moves tropomyosin out of the way, exposing the active sites on actin so the myosin head can bind. So we've covered this one. We will see this again. You're actually going to see this slide 2 more times. So we have to be hit by a nerve ending. We need an action potential and we need calcium. So what we're going to do is we're going to focus on this nerve ending first, and then in future parts when we hit the action potential, this one will be in bold, and then we will revisit the calcium even though we've already covered that part. And linking this electrical signal by a nerve to the muscle contracting is just called EC coupling. So again, we're going to focus on this nerve ending part right now. So we've already covered this, so I'm just going to move on, that the axons have to branch profusely. If you remember, every single muscle fiber has to be hit with a nerve. So there are 3 components of a neuromuscular junction, okay? The first one is the axonal endings. It is typically referred to as the axonal terminal. That is the end of the axon because it gets a little bit larger. If here is an axon as it's coming down, it's going to enlarge to increase the surface area. Inside these axonal endings, there are going to be these vesicles inside, and these vesicles contain a neurotransmitter, acetylcholine. That's the abbreviation for acetylcholine. So that's the first part. The second part, this is going to be really close to a nerve, to a muscle, sorry. And then that sarcolemma, right where that axonal terminal is, it's going to be really highly folded, and that's called the motor end plate. So it is a specific part of the sarcolemma. It is going to be highly folded. And in this highly folded area of the sarcolemma called the motor end plate, there are going to be receptors for acetylcholine. And then the third part is the space between the two, and that's called the synaptic cleft. All right, so these are the 3 parts: the axonal terminal, the motor end plate, and the synaptic cleft, the space between the two. So here we see this right here is the motor end plate. Right, it's highly folded, increases the surface area, and so we can put more of those receptors for acetylcholine. So basically what has to happen is we have an action potential travel down here. We needed to jump this synaptic cleft, start an action potential here, because remember what we've talked about previously, that once we generate this action potential here in this motor end plate, it's going to travel along the sarcolemma, down the T tubules, tell the paired terminal cisternae to release calcium. That calcium will bind to troponin, move tropomyosin out of the way, active site on actin is exposed, and myosin head can bind, muscle contraction occurs. All right, so we're going to go over exactly what happens as the action potential travels and reaches this axonal terminal. All right, because we have these neurotransmitters, these vesicles inside that have acetylcholine. We need that to be released, bind to these receptors, and these receptors are, once they open, this is the quick version, once they open, it will kickstart an action potential. All right. So once that nerve impulse is also action potential, once that nerve impulse reaches that neuromuscular junction, voltage-gated calcium channels open, calcium is going to enter. That will trigger exocytosis of those presynaptic vesicles containing acetylcholine. And so then that acetylcholine will be released into the synaptic cleft. It will diffuse across that synaptic cleft, bind to receptors at that motor end plate, opening those gates. Once those gates are open, and we're going to talk exactly how later on, but once those gates open, that's going to trigger an action potential. The other thing that we are going to need to do is we're going to need, as long as acetylcholine is bound to that receptor, the gate is open. We're going to need to close that gate. And so acetylcholine esterase is the enzyme that will destroy acetylcholine so that the gate can then close. So let me just back up. So as long as this acetylcholine is bound to this gate, it's open, and we need to close the gate once it opens. So once this acetylcholine binds, acetylcholine esterase will get rid of it, the gate closes. So this gate opens, and that's going to start this action potential that we would need to travel down the T-tubules, okay? And I believe I'm at a different place doing this recording, but